Here in this chapter of cell cycle, cell division and structure of chromosome, there are certain terms which are used. First we learn about what is chromosome. Chromosome is in the form of chromatin fibers which is present before cell division takes place, and it is thread-like structures. During cell division it becomes short and in a condensed form it is known as chromosome. When the cell division takes place, the chromosome consists of two sister chromatids which is attached at a point which is known as centromere. A chromatin which consists of DNA and histone proteins. This DNA, a double helix structure, is present in a sister chromatid. It is present in a sister chromatin. It is a double helix structure and it is coiled around histone proteins which makes up the chromatin. That is a chromosome. Chromatin becomes chromosome, and chromosome consists of two sister chromatids. Now these two sister chromatids each sister chromatid consists of double-stranded DNA. The double-stranded DNA, the two strands of the DNA are coiled around a protein which is known as histone protein. Chromosome consists of sister chromatid, two sister chromatids, and each sister chromatid consists of a double-stranded DNA. A DNA which the unit of a DNA is nucleotide. This DNA is coiled around a core. This is a core which is made up of 8 histone proteins. This complex is known as nucleosome. Nucleosome consists of DNA which is encircling or coiling around 8 histone protein, which is a core. This complex is known as nucleosome. So this is the sequence of DNA. So the whole DNA is formed in this manner. Therefore, the sister chromatid consists of DNA plus histone protein. Two sister chromatid make chromosome, and chromosome during cell division. The chromosome in a nucleus, when it is not dividing, is known as chromatin fiber, and chromatin fiber consists of DNA plus histone proteins. That is 40% of DNA and 60% of histone proteins. If we see the structure of DNA, the DNA consists of phosphate group, a pentose sugar, and a nitrogenous base. This is one side of the strand. The other side of the strand also consists of the phosphate group, the pentose sugar, and a nitrogenous base. Similarly, this is going to follow this is going to make one strand, and this is the other strand. Now there are 4 bases which are present: adenine, guanine, cytosine, and thymine. Adenine always pairs with thymine and guanine always pairs with cytosine. If we say what is present if adenine is present, then it is obvious the other side of the nitrogenous base will be thymine. If cytosine is present, the other side will be guanine. So this way the sequence is made. Now I have discussed about what is chromosome, what is chromatin, what is DNA, and now we come to what is gene. Here nucleotide, DNA is made up of nucleotide. What is a nucleotide? A nucleotide consists of 3 components. That is, which is already discussed earlier, a phosphate group, a pentose sugar, and nitrogenous base. This is one nucleotide. So the DNA consists of many units of nucleotide. A gene is few section of a nucleotide, of many nucleotide. Like this is one nucleotide, this is another nucleotide, this is the third nucleotide. So a gene is present and it consists of nucleotide, a section of nucleotides which consist of or encode. There is a word which is known as encode. Encode means it consists of certain proteins which is during cell division or in a offspring, it is expressed phenotypically. Phenotypically means it is expressed, it carries certain features. For example, tall, features like hair, features like eyes. So these are the genes which are present on a DNA, and it is a section of a nucleotide. It is few section of a nucleotide of DNA which is known as gene. Gene is only a set of nucleotides of a DNA.